chloroplasts only found in plants, right? They are double-membraned organelles. So we saw mitochondria, we saw nuclei, and now you're seeing chloroplasts, examples of organelles that have two membranes instead of a single membrane. And the inner membrane of the chloroplast encloses the aqueous fluid of the chloroplast called the stroma. And you see in this picture, you have these stacks. And this entire stack is called a granum. And each individual sac is called a thylakoid. It's actually the membrane of the thylakoid where photosynthesis takes place. So the electron transport chain that uses sunlight to convert sunlight into ATP and then use that ATP to make sugars. That's all taking place in the thylakoid membrane, and that's not the outer or the inner membrane of the chloroplast. So in a way, it's almost like there's a third membrane, right? So chloroplasts are pretty interesting in that way. Also, we still don't really know why chloroplasts have two membranes. We do know that there's a difference in permeability. The inner membrane is less permeable, so it's going to give tighter regulation of what goes in and out, and potentially that plays a role in increasing the efficiency of photosynthesis, but really we're just still trying to learn more. So already in our class we've seen a lot of examples of what we know so far, what we think, and what we're still trying to figure out. And in case I haven't mentioned it before, I want to make sure to point out that as we develop better tools to study organisms, we will continue to learn more. We also develop more different types of tests, but it's really having better tools, right? Like as we gain better resolution, we've seen more parts of cells. We've learned about texture. As we developed fluorescence and time-lapse microscopy, we've been able to see what cells do over a period of time. So all sorts of different ways in that different tools allow us to learn more than we were able to learn before. And so every time we get new information, there's always a chance that we will learn that we were wrong in our understanding before, and we will correct and update ourselves. And so science is not really saying like this is the end all know all, but this is what we know and understand so far with what we have to work with at this point in time. Okay. So make sure you memorize those key terms and you know the difference between the thylakoid, individual sacs, and the entire stack is the granum. Okay. All right, the central vacuole also only found in plants, and plants will have a large vacuole that occupies most of the area of their cell. As it fills up with water, it pushes the plasma membrane up against the cell wall, and that is what plants like. So though this might look a little strange at a glance, this is actually the normal state of a healthy, happy plant. When you see your house plants look droopy and sad, it's actually under the isotonic condition where there's no pressure being pushed against the wall. So plants actually like hypotonic conditions where water is drawn into the vacuole and it fills as full as the cell wall will allow it to fill. Okay, so plant cells have a large vacuole that occupies most of the area of the cell. It helps regulate water concentration under changing environmental conditions, meaning under hypotonic conditions, it's going to fill plump, plump, and that's going to be a happy plant. Under isotonic conditions, it's going to lose some water, and the rate of water going in and out is going to be at an equilibrium. Normally, this is what we think of as good, because for animal cells, we need isotonic conditions to be healthy and happy, but plants need hypotonic. So that's one big difference between plants and animals. And we will see this in a lab. We'll actually start to see it this week, and we'll emphasize it even more in coming labs. And then under hypertonic conditions, this is going to be detrimental to your plant. It's going to cause lots of water to exit and the membrane to pull away from the cell wall. And sometimes plants can recover from this if you add water to them. But usually this process is going to kill the cell. And if you kill enough cells, you'll kill the plant. Okay, so hypotonic conditions will lead to the cell expansion, right? It's going to fill up that vacuole and make the cell plump. The endomembrane system is internal membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and certain proteins. Okay, so we see the nuclear envelope, the lysosomes, the vesicles, the endoplasmic reticulum, and the Golgi Apparatus, and the plasma membrane, all are parts of the endomembrane system. The biggest two being the endoplasmic reticulum and the Golgi Apparatus. And if it needs to secrete anything outside of the cell, like to send it to other cells, for example, then the plasma membrane would be playing a key role as well. So here's a picture of that. So you have this interconnected system of membranes. It could be membranous sacs, it can be tubules, it could be vesicles, right? So here you see a vesicle, and it's going to deliver something to the Golgi apparatus. And here you see that right outside the nucleus, you have the nuclear membrane, and then directly after that is the endoplasmic reticulum. And so we tend to think of things going from the endoplasmic reticulum to the Golgi apparatus, and then either it's going to be used by the cell and stay in the cell, or it's going to be secreted out of the cell, and then you'll involve the plasma membrane. If it's a protein that needs to be kept in a vesicle, then it would take that from the plasma membrane. Most of the time, proteins don't need to be kept inside of a vesicle, so the vesicle will transport it to the membrane, and then the membrane fuses. The vesicle membrane fuses to the plasma membrane, and that's what will release the protein. And so the vesicles are just transports between the Golgi. Really, we have vesicles that transport between the endoplasmic reticulum to the Golgi, and then again from the Golgi to the plasma membrane. Okay, so why do we have all these? Well, the short answer is so that we can modify proteins, add, when you have complex proteins where you need to put subunits together, and maybe you need to add on non-protein parts to the protein, that's going to involve these steps. Also, all of the organelles in the cell have membrane coverings, and so you need to synthesize lipids and then deliver those lipids to the organelle to maintain their membranes, right? So there's a lot of reasons. So modify proteins through the rough ER, synthesize lipids in the smooth ER. The hollow portion of the ER tubules is called the lumen, and pretty much the hollow inside of anything in our body will be called the lumen. The membrane of the ER is continuous with the nuclear envelope. You already knew that. Okay. Ribosomes, when they're attached to the ER, it's called the rough ER. So ribosomes attach to the cytoplasmic surface to synthesize proteins. We do have some free-floating ribosomes that will also do this, but when when thinking about the rough endoplasmic reticulum, we're talking about the ribosomes that are embedded. And you can see a picture of that here, right? All of these red dots, those are the ribosomes. Okay, these new proteins are released into the lumen of the ER, and then they can be modified there. And so what kind of modifications might be starting the folding, right? Remember, proteins are going to be folded. Minimum, they're going to have tertiary structures. Most proteins even have quaternary structures, right? So you're going to start the folding process and any modifications that need to take place, acquisition of side chains and just additional parts to make complex proteins. Modified proteins are transported by the vesicles that bud from the rough endoplasmic reticulum membrane, and then they'll either be incorporated into cellular membranes or secreted from the cell. And one thing, remember we said it could bind and go to the Golgi and get further modified from there. It could bind to organelles and be delivered into the organelles, or it could bind to the cell membrane, the plasma membrane, and leave the cell. So protein hormones, that's very common, and some enzymes as well for being secreted from the cell. The rough ER also makes up phospholipids for the cell membranes, as I mentioned. We need to have a steady supply of phospholipids to maintain all of the membranes of all the organelles of all the cell organelles. And smooth endoplasmic reticulum, right, is also continuous with the rough ER, but you don't have the ribosomes attached to them. So here you see no red dots, no ribosomes. Okay, and the main role here will be the lipid, to getting the lipids from the smooth ER. Okay, so synthesis of carbohydrates can happen there too, lipids and steroid hormones. Also some detoxification steps of medications. This can be also where some of the medications can be processed and then become active. And then also you could have some poisons and detox those poisons there. And then there's also a calcium storing role that happens there. And so when we talk about calcium storage, there's a very specific role that we want to talk about. When you think about muscle cells, remember muscle cells do a lot of work and they need to be able to do this very quickly on and off. You need to have very fine, smooth control, motor control. And in order to react very, very quickly, both to contract and to relax, we need to be able to release calcium and then take it back very quickly. And so we have this specialized membrane called the sarcoplasmic reticulum in muscle cells. And so this is a specialized type of smooth endoplasmic reticulum. And it stores and releases calcium as needed for those coordinated contractions and relaxations of the muscle cells. So in response to an action potential, it's going to release calcium, and that's going to allow you to contract the muscle, but you don't want to be stuck with your muscle contracted, so you need to be able to take the calcium back, and then that allows the muscle to relax again. And so we are constantly doing this back and forth very, very quickly in our cells. We also don't use all of the cells in our muscles at one time, but we rotate through different muscles, which turning on some muscle cells at one time allows you fine control to exert more or less force. It also allows you not to fatigue instantly because you use all of your cells and they fatigue all at the same time, but you can let a few rest for a moment and use others and go back and forth and kind of cycle through. So there's a lot of very dynamic things happening and you think of your muscles being one, muscle group that you're using, but it's made of, billions of muscle cells that are all coordinating together, and calcium is quickly being released and then pumped back in to the sarcoplasmic reticulum to allow that muscle contraction and relaxation. Okay, and those individual units that allow for contraction are the myosin and actin fibers, which are located just below the sarcoplasmic reticulum, and they rely on the calcium. So in order for the myosin and actin to grab onto each other and contract, you need to release the calcium, and then you need to take it back to allow them to relax. Okay, the Golgi apparatus, we already kind of talked about it a little bit, but let's just talk about it again. So once you get through all of the endoplasmic reticulum, there's another set of membranes called the Golgi apparatus. And lipids and proteins within transport vesicles still need to be modified, sorted, packaged, and tagged so they get to the right places. And so that's what the Golgi apparatus is for. So you can kind of think of this as like your, when you're waiting for a package from UPS and it says it's reached a processing center. So you can kind of think of the Golgi apparatus as like that big processing and distribution center for all those packages. So everything gets delivered to the Golgi. It gets modified, sorted, packaged, tagged, and then sent out. A little bit more about the Golgi apparatus. So modification often involves adding short chains of sugar molecules. The modified and tagged proteins are packaged into vesicles that bud from the Golgi's transphase. So here, I don't think it's labeled, but you have the cis and then you have the trans. So they all come in on one side and leave on the other side. Some of the vesicles will be used elsewhere within the cell, and then others will fuse with the plasma membrane and be released from the cell and be delivered to other parts of the organism. Okay, cells with large secretory activity, like your glands, right? All of your glands are secreting hormones constantly to tell all the different parts of your body what to do. Or immune cells, they also have to secrete a lot of different substances to coordinate immune responses throughout the body. So those cells, gland cells and immune system cells, they're going to have a lot of Golgi apparatus material within them so that they can secrete all of their substances constantly. Plant Golgi bodies have an additional role of synthesizing polysaccharides. Okay, lysosomes. We already talked about the lysosomes in when we were talking about phagocytosis. So I can actually go back and show you this slide just so you know you're looking for this one. So the lysosome, remember, fuses to the vesicle that contains like the phagocyte, for example. And so the pathogen, you have a pathogen, it's contained in your vacuole, then the lysosome fuses and that delivers all the digestive enzymes. Okay, so that just came up again because the lysosomes are considered part of the endomembrane system. Okay. Cytoskeleton. The cytoskeleton is a network of protein fibers with several functions. So the first thing you think of is like our skeleton. It helps maintain our shape, right? The cytoskeleton also helps keep certain organelles in certain positions, certain locations, which is why you tend to see for different cell types, they tend to all look pretty similar, right? Because their general organelles are found in the same general place because of the cytoskeleton. And then this also allows movement of cytoplasm and vesicles within the cell. So remember you saw amoeba and how they were able to stick out those pseudopods and move. Their whole cell could move. That's using their cytoskeleton. And then, of course, this enables cells within multicellular organisms to be able to move. Okay. Now, 3 types of fibers within the cytoskeleton. We have microfilament, intermediate filament, and microtubules. We're going to have a separate slide for microfilament and microtubules, and this is the slide that we're going to use for intermediate filament. So intermediate filament is bigger than the microfilament, okay, and it's smaller than the microtubules. So you'll see it's the intermediate size. And an example is keratin. So we have the example here. Okay, so intermediate filament is only used for structural support. It is not being used for motility, okay? It helps the cell to bear tension, maintain shape, anchor the nucleus and organelles into place. So it is only structural. And keratin is the specific example, okay? Now, microfilaments, these can be involved in movement. They can be made very quickly and broken down very quickly, okay? So this could be to move the entire cell or just to move internal parts. It has the ability to allow movement of internal parts of the cell, or if the cell makes enough microfilaments in a particular way, it can use it to move the entire cell. Okay, so determine and stabilize shape. So it also helps to determine and stabilize the shape of the cell, and it's primarily made from actin. These little round, what looks like beads, those are all actin filaments. And then you just put a whole bunch of those together to grow your microfilament, and you break them apart to remove your microfilament. And this can happen very quickly, and that's why this is a good mechanism for motility for cells. Okay, the narrowest of the 3 fiber types, right? So microfilaments are the smallest of the 3 types, right? We saw microfilament, intermediate filament, and microtubules. Microtubules are the larger, and microfilaments are the smallest, and then intermediate is in between. Okay, so the narrowest of the 3 fiber types, just 7 nanometers thick. ATP powers actin to assemble its filamentous form, right? So to grow this chain, takes ATP to add on each bead to the next one, okay? And this is, actin is part of what we just saw for our muscles, right? Actin and myosin interact together to allow for motor movement, right? So actin specifically serves as a track for the motor protein myosin to slide upon, and that's your muscle contraction. Actin is also used for cell division and cytoplasmic streaming in plants. Microfilaments, a little bit more, right? Didn't all fit. They can depolymerize, so disassemble, or be synthesized very quickly, as I already mentioned. And this enables the cell to change its shape and move. Microtubules, so now the biggest of the 3 filament types. These are 25 nanometers in diameter. They are hollow tubes made of tubulin. So microtubules are made of tubulin protein. So it's kind of nice way to remember that. This does help form the rigid internal skeleton for some cells, provide the framework for motor proteins to move structures within the cell. and it's made of these dimers, so two tubulin proteins put together, and you'll get 13 chains of dimers surround a central cavity, and so here you see an image of that. So outer microtubule doublet, so the dimer, the doublet, two of them together, and then you get all those dimers, sets of dimers that go around, and then you'll have one located in the center. And that is actually how cilia and flagella are structured. So both cilia and flagella have this 9+2 array of microtubules. And I want to point out that this is eukaryotes. So there are flagella, some bacteria have flagella, but it's not this kind, okay? This slide is talking about eukaryotes only. And then cilia are only found in eukaryotes, okay? Cilia are shorter, and you're going to have lots of them, and they're going to beat together in particular patterns to provide the function. So when you think of the cilia that line your lower respiratory tract, they propel phlegm or mucus up and out of your lungs constantly, which is swallowed, and you're not aware of it. You don't feel it. And this helps to clear all of the stuff in the air that's constantly getting into your lungs, right? Dust particles, viruses, bacteria, pollen, all sorts of particles in the air. You don't want those sitting in your lungs and interfering with your ability to exchange oxygen and CO2, right? We want to get CO2 out of the blood. We want to get oxygen into the blood. And we want clear membranes to do that. And so mucus lines your respiratory tract, traps all those particles, and then is moved up and out. Is there anything else I wanted to say? Flagella of eukaryotes are coated in a plasma membrane, and they are more complex than flagella of bacteria. Not all bacteria have flagella, but those that do, it's a simple form of flagella. Eukaryotes, again, only some eukaryotes have flagella, but those that do, it's much more complex and is coated in a plasma membrane. All right, extracellular structures. We have plant cell wall, and you know the plant cell wall is made up of cellulose. It provides support, barrier to infection, and it also allows for connections to connect adjacent cells. And plasmodesmata are these special connections, and those are only found in plants, right? Then in animal cells, we have the extracellular matrix that's made of collagen and other fibrous proteins. This helps with various activities, one of which is just to be able to form tissues, to have cells stick to each other. There's glycoproteins called proteoglycans, so proteoprotein, glycan, sugar, so proteins and sugars, okay? And then there's linking proteins as well. So we have this very diverse, I guess you could say, matrix that is outside of the cell. The cells will excrete these materials. They'll usually be anchored somewhere along the membrane, and these will carry out different roles for the cell. And we have, as we mentioned, the plant junctions or connections. Well, animals have junctions or connections as well. And in animals, we call them tight junctions, gap junctions, and desmosomes. And they have specific, distinct roles, and you need to know each of those. So we're going to talk about that. So for plants and animals, these direct channels between cells allow them to communicate and allow for other special functions as well. So we mentioned between plant cell walls having these special channels on plasmodesmata, and these are channels that pass between the cell walls and plants and allow the cytoplasm to be shared between adjacent plants. And this is one way that plants can move materials from one cell to another cell. They also have vascular system, xylem and phloem, where xylem moves water throughout the plant and phloem moves nutrients throughout the plant. But for other sharing of other materials between cells, they can use the plasmodesmata. Now, animal cells, the tight junctions, these are watertight seals, so they can prevent anything from leaving that type of tissue. This is incredibly important in our digestive system, where we don't want everything that we ingest to just go straight into our bloodstream. We want to select what we allow to go into our bloodstream to keep ourselves safe. So we have tight junctions that line the entire epithelial of our intestines, right? And we also have this on our skin, right? We don't want our insides leaking out, right? So, and our internal organs, other organs are also going to have this as well. So tight junctions, super important, watertight seals between the cells. And these are found in animal cells specifically. Desmosomes are like really strong grips. These join adjacent cells, especially important when you think of skin or like muscles, hearts, lungs, things that need to stretch constantly. Like you take a deep breath, your lungs expand, and you exhale and they retrieve, right? And your skin, obviously, you're able to move, stretch your skin. Muscles, right? When you contract a muscle, they bulge in one area where they weren't before. So tissues need to be able to be flexible and move. But you also don't want all the cells just moving around freely and being disrupted when you do these activities. So we have certain locations between cells where they're attached very tightly, and then the rest of the space between those cells is kind of flexible and allows for movement. So desmosomes are only found in animal cells. They're short proteins that extend from the plasma membrane of one to the plasma membrane of the adjacent cell, and then they're like spot welds, like permanent, very strong attachments, okay? And these adjoin cells in things like skin, heart, lungs, and muscle. Okay, and then lastly, we have the gap junctions. And the gap junctions are going to act as just channels to pass materials between adjacent cells. And this is particularly important in cardiac muscle, where you need to be able to have that coordinated contraction and relaxation between the ventricles of the heart to keep blood moving in one direction. And so this helps share materials that are involved in the contraction of the muscle. Okay. So gap junctions are made of 6 proteins called connexons, and they form an elongate, like a donut that you stretch out and is elongated to make a channel. Okay. And when those one elongated donut-like structure called a connexon with an O is connected. So you have one end in one cell that makes a channel, and then the other end in the adjacent cell. And so the space here is space between two cells. This is one cell, and then down here is another cell. Okay. So when connexons of adjacent cells are aligned, it completes the channel. And so you can see kind of in between where it can be closed, and it can when it's lined up, it completes the channel. And so this is open and it's closed. So there's control, and it allows, these are not permanent attachments like we saw with the desmosomes. Okay, these are able to be closed open. They're able to form and reform. Okay, so we have now finished talking about the eukaryotes. So make sure you review, compare and contrast prokaryotes with eukaryotes, and then also make a compare and contrast chart for your plant versus animals. Okay, so this has a lot of information on it for you. But again, this is to get you started. I want you to go through as you study, continue to add things, and then again, make sure you make one for plants versus animals. Also, make sure you know the different carbohydrate cell wall compositions of plants, fungi, and protists, which are all eukaryotes. Okay? And then also, I put this here so you could practice memorizing the different parts of the cell. This is an animal cell. You can do the same for your plant cell. So here's a blank one for you to practice filling in the blanks. See if you can first name all the organelles and then describe what the function of the organelle is. I gave you also the answer key, so you can check your work. So what I would do is print out a couple of these so you can practice, and then you can check your work against the answer key. Okay? And then your homework here, which is already posted on Canvas, and some of you already did it, which means you've already done the reading and you've already started studying, which is great because your exam is next week. But hopefully you also are listening to the lectures, because there's a lot of information in a lecture that's not necessarily written on the slide or in your textbook. All right. Email me if you have any questions. Good job.